Hello and welcome to the Fluidine's Field Application Scientist Friday session for May 15th. My name is Anita Khan and I am a mass cytometry FAS supporting customers in Northern California, the Pacific Northwest, and Western Canada. I'm joined by my colleague, Lindsay Rapkin, who is an imaging mass cytometry FAS supporting customers in the Northeast. We'll be taking your questions in about 15 minutes or so. Please feel free to enter the questions in the question box at any time, and we will answer these questions after the presentation. So first, we would like to discuss how mass cytometry and imaging mass cytometry are leveraged in many key research areas. Mass cytometry and imaging mass cytometry are used across a broad range of applications. Some of these research areas are immunology, infectious disease, immuno-oncology, autoimmunity and allergy, and oncology and a tumor microenvironment, or TME. In the next few slides, we will discuss publications where mass cytometry and imaging mass cytometry were utilized in some of these key application areas. Due to time constraints, we will not be able to cover all applications in great details today. Please don't hesitate to reach out to the mass cytometry or imaging mass cytometry FAS in your area to learn more about our technology and how it can be applied to your specific research questions. So first we'll discuss some key research applications using mass cytometry. So in this paper from Jim Ellison's lab at MD Anderson Cancer Center, distinct cellular responses to combination anti-CTLA-4 plus anti-PD-1 checkpoint blockades were identified. Mass cytometry was chosen as the sole method to investigate cellular responses to either dual immunotherapy or monotherapy. Murine tumors and peripheral blood from melanoma patients were assessed with 40 plus marker CyTOF panels. Metaclustering identified 14 tumor infiltrating T cell subsets differing in frequency between mono and dual immunotherapy conditions. Combination therapy and anti-CTLA-4 therapy, but not anti-PD-1 therapy, were found to elicit a switch from expansion of exhausted CD8 positive C cells to an expansion of activated effector CD8 T+. Switching on to some scatter measurements. Diagnosis of hematopoietic malignancies is traditionally done by IHC, immunohistochemistry, or by flow cytometry. IHC is a skill-based technique and is variable, while flow is limited by the number of markers and autofluorescence of certain cell types. In this publication, Sean Bendall's lab from Stanford developed a morphometric analysis for mass cytometry to construct two-dimensional visualization equivalent to forward and side scatter measurements used in flow cytometry. So this approach combines markers that can quantify the morphological features like granularity, cell size, granule color, chromatin quality, nuclear shape, nuclear size, cytoplasmic color, referred to as scatter bodies, as well as the traditional CD phenotypic markers and a reproducible dimensionality reduction technique that is based on linear discriminant analysis. See, now mass cytometrists have access to and can quantify features that pathologists can recognize after multiple years of training. This method can potentially identify unique diagnostic markers as well as myeloid differentiation pattern for normal and neoplastic blasts and clonality that a pathologist cannot identify via traditional microscopic or flow cytometric methods. So coming to type 1 diabetes, in this publication from Alice Long's lab at the Benaroya Institute of Allergy and Immunology, A 33 marker mass cytometry panel was used for deep characterization of autoreactive CD8 positive T cells in type 1 diabetes patients. The panel also included tetramers to identify islet-specific CD8 positive T cells. So islet-specific CD8 positive T cells with an activated memory profile were to be associated with rapid progression of type 1 diabetes, while CD8 positive T cells with an exhausted profile were correlated with slow progression of type 1 diabetes. So these findings may lead to biomarkers of disease trajectory and response to treatment, as well as possibility of new targets for therapeutic intervention. Mass cytometry is widely used in mouse models also. In this paper, the Rolls lab at Technion Israel Institute of Technology used mass cytometry to perform a high-dimensional single-cell characterization of immune cells in the naive mouse brain. Use of a 37-marker CyTOF panel enabled broad analysis of immune cell subsets in the immunological milieu of the murine brain. Previously undescribed immune populations were identified, including some rare subset with frequencies of less than 1%. The study also demonstrated the complexity of immune population in the brain. This comprehensive map of brain immunity provides identification of specific cell subsets relevant for further disease or therapeutic studies. A recent study of COVID-19 patients undergoing treatment for inflammation symptoms caused by the virus shows the power of mass cytometry to visualize both phenotypic and functional changes across the immune system as a whole over time. This enabled a system-level analysis that showed overactivation of T and NK cell subsets that likely contributed to inflammation in the severe cases. The researchers did not have to guess at which populations to focus on before beginning their study. Mass cytometry provides the breadth and depth needed, especially when investigating a new disease. Profiling the human immune system is a key application used in translational and clinical research to look for biomarkers of disease or response to therapy. It is essential to driving the search for more effective treatments for almost any disease: cancer, autoimmune diseases, infectious diseases. The MaxPar Direct Immune Profile System is a truly revolutionary approach to immune monitoring and is absolutely unique in the cytometry market. The researcher just adds a small amount of blood to a lyophilized pellet of 30 antibodies in a single tube, processes it, collects the sample on a Helios mass cytometer, and performs data analysis with specialized software that produces quantification of 37 key immune cell populations in minutes. So let's switch gears. Let's discuss about some key research applications using imaging mass cytometry, or IMC. This slide shows a recent report from the Annals of Internal Medicine. The authors used an 18-marker imaging mass cytometry panel to enable an unbiased exploratory approach to assess immune infiltration in lung tissue from 2 COVID-19 patients, one who died of cardiac arrest and one from pneumonia complications. Differences were noted in the immune patterns of infiltrating CD4 positive and CD8 positive T cells, NK cells, and macrophages in a single section of lung tissue from each patient. Recruitment of aberrant CD45RA positive T cells is an immunologic feature of COVID-19. Once bacterial pneumonia occurs, some phagocytes recruited by CD4 positive T cells begin to play a significant role in lung injury. In this paper from Bernd Bodenmiller's lab at the University of Zurich, a 37-marker IMC panel was used to define 57 breast cancer phenotypes. Breast cancer genomic subtypes were also found to exhibit marked differences in stromal phenotypes as well as cell-to-cell interaction. IMC-derived cell phenotypes were linked to clinical outcome, potentially identify tissue biomarkers, and reveal prognostic effects that are dependent on genomic context. Imaging mass cytometry and spatial organization were fundamental aspects in this paper from laboratory of David Ream at Yale University School of Medicine. Here, they compare imaging mass cytometry to their previous quantitative immunofluorescence studies and show that the data correlates well. In addition, the Hyperion imaging system allowed the researchers to expand their panel of markers, enabling the examination of the spatial relationship between HER2 expression on either the intracellular or extracellular membrane of tumor cells and the presence of CD8 positive T cells. They report that the increased proximity and levels of cytotoxic T cells, especially to cells expressing extracellular HER2, correlate with improved responses to treatment with either one or a combination of the two immunotherapeutics. The authors claim that the multiplexing capabilities of the Hyperion imaging system may help to identify patients that derive benefits from targeted treatment or immunotherapy. In this paper from Fritz Koning's lab at Leiden University Medical Center, the authors report how they developed a 40-marker panel for high-dimensional characterization of cancer immune microenvironments by imaging mass cytometry. This panel for FFPE tissues included immune cell markers to assess tissue residency, lineage, and activation phenotypes. In addition, the panel included surrogates of cancer cell states as well as tissue-specific markers for cellular contextualization within the tissue. The authors also describe an optimized workflow for maximum antibody performance as well as providing insights into the antibody validation process. Imaging mass cytometry is also used for investigation of tissues other than tumors. The papers listed on this slide, one from Bernd Bodenmiller's lab at the University of Zurich and the other from Klaus Kessner's lab at the University of Pennsylvania, studied the pancreas and disease progression in type 1 diabetes. Key findings of these papers included identification of significant alterations in pancreatic islet architecture, endocrine cell composition, and immune cell presentation, as well as identification of cytotoxic helper T cell infiltrates in beta cell rich islets and loss of beta cell markers preceding beta cell destruction. So in the final imaging mass cytometry example today, Jennifer Gomerman's group at the University of Toronto leveraged the multiplex capacity of imaging mass cytometry to ascertain the phenotype and location of immune cell subsets in the post-mortem brain of a patient with an acute form of multiple sclerosis. Being able to visualize many different markers in a single sample enabled the authors to identify subsets of T and B cells and distinguish resident microglia from infiltrating macrophages. The spatial context of the image allowed the authors to determine which types of lymphocytes are in close proximity to blood vessels. This finding provides insight into how cells move through the brain and the order in which the cells may arise at the site of inflammation or injury. Not only did the author state that the imaging mass cytometry results are consistent with previous findings based on immunofluorescence, but that the Hyperion is a powerful tool to further our understanding of the inflammatory response and tissue injury in the multiple sclerosis brain. So we'll stop here and we will take some question and we'll answer your questions in further. So thank you for your attention.
Thank you so much, Anita. You presented some amazing work here. We are now happy, as Anita said, we're now happy to answer your questions. So please enter your questions in the question box and we'll try to answer as many as we can. The questions that can't get answered during the webinar will be forwarded to your regional field application specialist and they will follow up with you directly. Oh, great. I'm starting to see some questions coming in. Excellent. Anita, the first question is for you. And the customer said that we are working with tumor samples, but we think a CD45 enrichment would be needed for better counts. Would the magnetic beads cause problems with mass cytometry?
Thank you, Lindsay. That's a great question. So beads used for CD45 enrichment, like from those from Miltenyi, they can be used as long as they are smaller. The diameter is smaller than 100 nanometers. So that should be fine. Anything larger cannot be ionized in the mass cytometry. It's a mass spec based on ICP MS. So we won't be able, the mass cytometer needs beads which are 100 nanometers or below. And make sure, that's the most important thing, make sure that if you're using such bead preparation, they're not contaminated with other heavy metals or impurities. So you can run a very, very diluted amount of these beads in the mass cytometer, and then you can check if there is any contamination. You can also use nitric acid to dissolve the beads and then run that in solution mode instead of suspension mode. Your FAS, work with your local FAS and they will help you go through this procedure. So making sure that your cytometer is not contaminated by heavy metals which might be in these preparations. So I hope you can work with your local FAS. Thank you.
Great, thanks, Nita. The next question is an imaging mass cytometry question. And it says, could you please describe the IMC staining protocol and is it complicated? The staining protocol for imaging mass cytometry is actually quite similar to that for IF or traditional immunohistochemistry. The main difference is rather than using fluorophores with our antibodies, we're using metal conjugated antibodies. The rest of the protocol is quite similar with a rehydration step, antigen retrieval, and then you can actually stain all of the antibodies in your panel simultaneously, which saves a lot of time in your protocol. We have protocols to help support you with it, so if you have any further questions, again, please reach out and we can help you with every step of the way. We've done the protocol many times. It's actually quite simple. The second question is another one based on immunofluorescence or a comparison, and it says, what are the advantages of using imaging mass cytometry to a standard technique like IF? IF makes use of fluorophore conjugating antibodies, and while they're great for a technique like IF, they do have limitations for high parameter studies. Some of those limitations include signal overlap or variable staining intensities, or even your tissue might have background signal or autofluorescence, and it's inherent in your tissue and it can interfere with some of your signals. And because of this, traditional IF is restricted to only a few markers at a time. Imaging mass cytometry can overcome a lot of these limitations by using metal tags conjugated to our antibody. These metal tags result in separate and distinct signals. They have uniform staining intensities, unlike fluorophores, and are not naturally found in biological systems. And so we don't have any issues with background or autofluorescence. So the next question looks like it's going to be another question for mass cytometry. Anita, this is for you. Is MDIPA only for PBMCs and whole blood? What about tissue section?
Oh, that's great. So we have definitely validated the panels, that MDIPA panel for PBMC and whole blood. And there is a lot of literature available on our website. The protocol, everything is available. But if you're thinking about TILs or the the immune cells that you take out from the tissues. You may have to optimize it, you may have to check it, because some of the clones may not be compatible with the fix and perm conditions. So you may have to do some optimization on that part, but we have not validated on tissue infiltrating lymphocytes or on that. So definitely, please do work with your local FAS to see if this is feasible. Thank you.
Great, thanks, Nita. The next question that came in is asking about analyzing IMC data, and specifically they said, could you highlight some of the steps in analyzing your imaging mass cytometry data? Sure, so to answer that question, we'll start with a little bit about our pipeline. We have it in 3 steps. Once you have your data acquired, we take a look at it using our MCD software, and then the next step is to segment our images. And cell segmentation is an analysis technique, and it's required to identify your cells on an image. And this is essential because it's going to allow us to perform quantitative single cell image analysis on your data. Once we create this segmentation or this mask, we can upload it to an analysis software and along with original images. And then we can perform higher order analyses where we can visualize or plot your single cell data. You can cluster your data into distinct populations. We can gate on certain cell types. We can go back and look at these populations and gates in your original image. Ultimately, at the end of the day, there are many possible analysis routes that you can take, and which one you choose depends on your specific research questions. And different labs are constantly modifying these techniques and making them work for them. But a great place to start is looking through the literature, looking to see what people are doing, and of course, contacting us. We can help you with our pipeline from getting your images and your panel design right along to doing your analysis and helping with generating publication quality images. The second question seems to be a follow-up to that one. And it says, do we need to purchase additional software for the Hyperion image analysis process? The instrument control software for data acquisition is included with the purchase of the Hyperion. And so you won't have to, if you are operating the instrument, then you'll get that. For users, you will work with the operator and you can take a look at your data. In terms of segmentation and data analysis, we support free open source versions. For segmentation, we can use Cell Profiler. And for data analysis, we can use Histocat, which is also free and it's available online. There are commercial software choices if you're interested, such as Halo or Visiopharm, which Fluidigm does have co-marketing agreements with. If you're interested in any of these software options, we can help connect you. But we have support for Histocat, Cell Profile, and any of the other data analysis questions you may have. So if you have specific questions, again, reach out to your FAS and we'll help you along that data analysis path. Okay, the next question is for Anita, and this customer is saying, can you label streptavidin with metal tags for tetramer detection?
So that's great, because a lot of people are now very much interested in tetramer, especially with respect to COVID and detecting the T cells which are involved in involved in COVID protection. So a lot of people are asking that. Streptavidin on its own cannot be labeled straight to the metal. It needs they need more cysteine residues. So there is a recombinant cysteine streptavidin which is available, which is used for metal binding. We are definitely will be happy to point you towards Evan Newell's publication describing the procedure and the original cysteine recombinant cysteine streptavidin paper too. So you can check out if you want to follow those procedures. If you are interested in tetramer research in your lab, work with your FAS. There are commercial level tetramers that are already labeled with PE and APC, streptavidin labeled with PE and APC. You may want to use that you want to use your tetramer and then the streptavidin, PEAPC conjugated streptavidin, and you can use fluidines metal conjugated secondaries against these dyes to actually detect. That might be simpler. And please refer to online fluidime catalogs for these secondary antibodies, and work with your local FAS to standardize the protocol, to optimize the protocol. That will be my advice at this point. Thank you.
Thanks, Anita. There's one more question, and it's for you. And it says, what are scatter bodies?
Yeah, the scatter bodies that I mentioned in Sean Bendel's paper, these are the morphological markers. There are 11 markers he used, and then also barium that would go for granularity or granular color, chromatin quality, nuclear shape, nucleolar size, cytoplasmic color. Basically, the morphological markers that will distinguish different cell types. For example, he uses VAMP7 for vesicle docking, or MPO for granularity, lysozyme again for granularity. So these are laminin, lamin B1, HP1. These are some of the markers I remember on the top of my head, but these are the morphological markers that were available classically by flow cytometry because it has side and forward scatter. But now using the antibodies against these markers, which are surrogate for these morphological markers, they can actually now not only replace those forward and side scatter measurements, but actually they can quantify it. So that is why these are called scatter bodies. And then he also adds barium so that the morphological or morphometric analysis is very powerful. Thank you.
Thank you, Anita. Another question came in for IMC, and it's saying, should we cluster analyses per sample or cluster analysis all samples together to identify subgroup of cells? That's a good question. The best way to answer is to say it depends on your question. We have some researchers who tend to do their clustering separately and then to identify their groups, their subgroups that way, and then come and compare them to their other subsets. We have others who tend to put them all together. I think it ultimately depends on your question and what you're trying to answer. So the best way, again, is if you have a specific question in mind or you have an experiment where you're toying with how best to do this, contact your FAS and we can work through your question and see what the best way to answer this would be.
I have a follow-up on that for mass cytometry. See, if you are having data sets from one set of sample, let's say a clinical trial, you have taken it month 1 and then another set after 6 months, and those samples are then run, and then they're trying to analyze and make sense of those two data sets, one from month 1 and one from month 6. Please do include proper control samples within that. So what happens is that when you do clustering together, there is a common control so that you can check out if things are whether you can compare those things, because sometimes month 1, month 6, if you have done the staining separately, there might be issues with your staining technique, there might be issues with the samples themselves, there might be issues with the reagent may not have been stored properly or may have precipitated antibodies. A lot of these things can happen between month 1 and month 6. So you want to make sure that you include a common control which can give you some ideas. Otherwise, the for unsupervised clustering especially, the patterns won't look very similar, and that has happened before. So including right controls in your in your samples is extremely essential, as it is essential for any other scientific experiment.
Thank you, Anita. So we're getting to the end of our webinar today. So if you have any additional questions or you want to follow up or talk about any of our technologies, You can always schedule a one-on-one with a field application specialist. In North America and Canada, you can send an email to support.northamerica@fluidigm.com, or if you're in the MEA, send an email to eu.support@fluidigm.com. Please join us for these upcoming webinars. On the left side, we have topics on mass cytometry and imaging mass cytometry. On the right side, we have a spring microfluidics webinar series showing the last 2 of the 4 series remaining. You can see the complete list and register at the fluidigm.com events page. Here we have one more topic on mass cytometry and imaging mass cytometry. As a reminder, join us each Friday for FAS Friday Q&A. If any of your colleagues missed today's session, we'll have a repeat on June 19th, so please don't forget to invite them. Again, all of these upcoming webinars are listed on our fluidigm.com events page. Please visit and register there. Finally, we'd like to mention this brand new session to introduce the Advanced AccuLift Laser Capture Microdissection System. You can choose the date and time that works best for you in your time zone. In addition, next week's FAS Friday Q&A will feature this subject. We hope to see you there. Again, thank you for joining our webinar today. This will conclude our webinar.